Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. Today is September 12th, 2024, and we have with us Brian Lundstrom. Brian is an associate professor in neurology and biophysics in the Division of Epilepsy in the Department of Neurology at Mayo Clinic in Rochester, Minnesota. His work is at the intersection of diagnosis and treatment of brain diseases, experimental neurophysiology, biophysics, and theoretical neurobiology, which spans kind of a large spectrum, and we probably won't get to all of that, but maybe. So welcome, Brian.
Thank you.
Also with us today is our own neurophysiology experimentalist and theorist, Fidel Santamaria.
Hello, Charlie.
Fidel. And me, I'm your host, Charlie Wilson. So Brian, your work on epilepsy is a great example of bioelectronic medicine. It's a phrase you used, and we'll get to it, I think. The problem with epilepsy is that it's a clear case of a disease of brain electrical dynamics, and it's a kind of circuitopathy that needs to be treated using electrical treatments, diagnosed using electrical diagnostics. It is an electrical dynamics disease. And so could you start by reminding us the state of electrical recording in diagnosis and treatment and care of patients in epilepsy?
Yeah, epilepsy is defined by seizures, of course, and seizures are something we can see very well on EEG, or a way that we record activity from the brain. And EEG has been used for almost 100 years to record that kind of activity, and for a long time for diagnosing and more precisely categorizing epilepsy. The treatment of epilepsy has long centered around medications and then resection and destructive procedures. But more recently, brain stimulation has become much more commonly used. And in some ways, you can think about medicine as being comprised of medications that we might give or surgeries that might be performed, but bioelectronic medicine, or using electricity to treat a disease, is something that's been done for a long time, but is more commonly being done and better understood now than ever before. And so at Mayo Clinic, I'm an epileptologist, so I see epilepsy patients and help treat them with any of the ways that we can do that. But I also then specialize in brain stimulators. So using brain stimulation to treat epilepsy. And I have a background in computational neuroscience and a long-standing interest in understanding how neurons communicate information and themselves maybe control their excitability, or how gain control is accomplished by neural networks in the brain. And so some of my research then in some ways sort of mixes these two interests, that of epilepsy, treating the epilepsy with brain stimulation, better diagnosing it and tracking cortical excitability with EEG and epilepsy, and then, of course, hopefully better understanding the brain and the process.
When you diagnose a case of epilepsy, you're usually looking for a place in the brain where the seizures are starting. And that's normally done just by recording EEGs all over the brain and looking for the place where it's biggest? Is that the idea, or where it happens first?
Yeah, the general approach is that when we first see patients with new onset seizures, we want to first categorize them into generalized onset seizures or focal onset seizures. These days, that would fall into a category of focal epilepsy or what is sometimes called genetic generalized epilepsy. The reason that's important is because it changes how we treat the patients. If it's focal epilepsy, which the majority are, then we're looking for an area of the brain that we could potentially treat. Now, 2/3 of patients will be successfully treated with medications. Although they do carry side effects, they can be very effective. But for other patients, we're looking for other ways to treat them. And one of the key ways that we decide what is reasonable is by, as you mentioned, recording seizures on an EEG. And when we record seizures on an EEG, we first start with the scalp EEG. That's the easiest. And we're looking for where seizures start. We can also look for interictal discharges, so discharges that we see on the EEG that happen all the time and are strongly correlated with hyperexcitability and seizures. And the location of these interictal discharges can give us an idea about where the epilepsy is coming from, but not to a very specific degree. It's a more general idea of the location. Recording seizures on scalp EEG gives us a little bit better of an idea. And then, if we need a better idea than that, for example, because medications aren't working, we might at some point even recommend invasive EEG, where EEG electrodes are placed inside the brain, and we can better localize seizures that way.
So you can't just do a scan, a MRI or something, and see where the seizure.
Well, we always do MRI scans if we can, and it can be helpful. So some patients have what's called lesional epilepsy, where we see where possible lesions are located. We still want EEG to help confirm that, but there's quite a few patients who are non-lesional. And that means that their MRIs are unremarkable, and so that doesn't help us.
So something's going wrong, and you can't see anything.
I think it's a general principle that that what we can see on MRI and what we can detect if we're trying to measure brain function with the state-of-the-art ways that we assess brain function is very crude in a lot of ways. There's a lot of abnormalities that exist on MRI. Well, let me say that exist in the brain that you cannot adequately see on MRI. And so it's a little bit of a misconception sometimes that if you have a normal MRI, it means everything's okay.
But what about a fMRI? What if you just look for like the bolt signal, and then you will just following the intuition, I guess, that there are neurons that are more prone to be excitable, and you will imagine that they will consume more energy. And is that a way?
Yeah, people have tried EEG linked fMRI. Mm-hmm. So you basically will find EEG, these interictal abnormalities, and then you'll look at the bold changes related to that. And that can be helpful. Maybe more commonly used is FDG PET, positron emission tomography, where you're really looking at metabolism, right, and you're looking at actually areas of hypo or reduced metabolism, and that can help us localize the seizure activity. But these are all, relatively speaking, still crude measurements that don't remove the oftentimes necessity for invasive EEG.
Also, they're sort of indirect. I mean, what's wrong is the electrical activity, and that may alter energy consumption.
That's right.
You have a measure of electrical activity.
Yeah, that's right.
Measure that.
Yeah, and this is an interesting thing when you think about EEG. EEG's been around since the late 1920s. And for a long time, EEG was the way to, quote, image the brain. Until CT and MRI were here, it was the, quote, brain imaging approach of choice. And then after that, it really became restricted in its use to epilepsy and seizures only. Now, interestingly, since maybe the early 2000s, it has been increasingly used in the hospital for severely ill patients for long-term monitoring, where things may be going wrong and there may be subclinical seizure activity or activity that is not clearly seizure by what we can see, but we can see abnormalities on EEG, and so it has been used in that way more. But broadly speaking, EEG is still read by just looking at the raw signals, and it's not particularly sensitive for all the neurophysiological complexities that we know exist. So it's probably an area of untapped potential still.
So we just can't look at it and know what's going wrong. I know what's wrong. These neurons are too active and these neurons are not active enough. It just doesn't give you that kind of resolution.
Well, we don't actually know to some extent whether it can or can't, but I think for sure it can't in the way that we currently use it. And that is the clinical standard of care. We read raw EEG. I mean, nobody reads raw fMRI signals. That's all relatively highly processed. But the problem is that we don't really understand how to appropriately process EEG in a way that's better. So we just.
But it is like it's a problem. I mean, EEG. I mean, like from the little I remember, you can have multiple sources, and then that will generate the same signal. And there was a discussion at least like 20 years ago, like, oh, how many poles do you have?
Yes.
And like there was this kind of 8, like people will say, and then that explains the EEG signal in the entire brain. But that and their location is not. It's a very kind of degenerate problem. And
yeah, from a research standpoint, that's exactly true. There is a whole aspect of EEG related to what's sometimes called ESI or EEG source imaging, where the idea is if you take a discharge, you would like to localize where it came from. But that problem has many solutions, and an infinite number actually. It's sometimes called the inverse problem, and it's just an ill-formed problem. And so that remains a problem. But even more broadly speaking than that, the big problem with EEG is that we're looking at a spatial scale that is very large. And we're trying to learn something sometimes about a much smaller spatial scale. And how we move between abnormalities in neurons and even small networks and what we see on EEG is unclear. For example, there is a long history in reading EEG where people would pick out certain waveforms on EEG, they would give them a name, and then they would try to associate that with a disease. And the idea was, well, if we see enough patients with a particular disease and they all seem to have this waveform, then maybe we can just correlate them. The problem
is strictly descriptive, right? It wasn't
That's right.
based on. It's not like that.
There was
EKG where you know what each of the bumps.
That's right.
Like the evoked potential. I mean, in the evoked potential, there was like alcoholism and things like that. In the evoked potential, the signal decreases, and that could be related to EEG. But
yeah, but broadly speaking, Charlie, you're exactly right that there wasn't a mechanistic connection between the disease and the EEG findings in general. It was just sort of association, and really a lot of that, especially once we started to do more long-term monitoring in the hospital, has changed. And the reason it changed is because it was never that helpful or accurate. Right. And that may not be surprising when you think about it. If you think a complex disease that is likely very heterogeneous, do you really think that it's just going to have one single pattern on the EEG? I mean, deeply thinking about it, it doesn't necessarily seem like it's going to make sense. What you need is some better framework of understanding brain physiology. That's probably what's reflected in the EEG. And so you need some sort of framework to kind of connect mechanistic understandings of disease with how that might affect the EEG within what we expect to see on the EEG. So interestingly enough, just to give you an example, what has been done in EEG is instead of trying to associate EEG waveforms with specific pathologies, more recently people have tried to be a little bit more careful about just associating specific waveforms with whatever terminology we can come up with, which clearly describes the waveform. And then we can do some more careful scientific studies that try to associate that with underlying causes. Any of us who read EEGs can recall these sort of almost comical discussions that you might have where you don't want to call a waveform, for example, you don't want to call it a triphasic wave because you think the patient actually needs an anti-seizure medicine, although what you see on the EEG is clearly a triphasic wave. But if you say triphasic wave, you know that the person interpreting the EEG will think that they don't need an anti-seizure medicine because they've always been taught that triphasic waves are benign. So this is this kind of circular logic. You're like, I'm not going to call on the EEG what I see because I think the person who reads it is going to do something that I don't think they should do. So this is a very tricky thing, and we really need to get away from it. And so that's sort of what's been done. But at a more scientific level, what needs to be done, I think, is we just need a better way to bridge the spatial scales from some of these abnormalities and this excellent fundamental neuroscience that is going on in a lot of places and what we can see on the EEG as a result.
So where do we stand on that? Is there a way to kind of zoom in? So you mentioned invasive EEG, which means that you could be looking at a smaller spatial scale. Is there a way to sort of continuously zoom in, which would allow you to see the same event at different scales and identify it as the same event?
It's a good question, and I guess the answer from my perspective is we don't know for sure clearly. With invasive EEG, we certainly see a smaller spatial scale. But we still see waveforms that are a little bit unclear, and we don't know how that relates to still some of the underlying physiology and pathophysiology. On a research level, we, and as well as other places around the country, do some microwire recordings where we can record multi-unit activity and sometimes even single-unit activity from humans. But correlating that with exactly what's going on at the neuron level and the neural network level remains difficult. So I think there's still, unfortunately, a relatively large gap still.
But you could relate features or properties to certain propensities, right? I mean, not to the to a biophysical level. That's very complicated, and maybe will that be that useful? I mean, maybe to something about telling you about the health of a circuit as a metric that tells you at least this overall area or in this time of the day, this functional circuit is in this condition that is more prone to a seizure or not. Is that?
Yeah, I think that's sort of where we need further work, and maybe you could, in some cases anyway, call this a mesoscale, where maybe there's EEG signatures of what is a healthy neuron. If it wouldn't be an EEG signature necessarily, but a healthy neural network maybe. And for microwire recordings, what do healthy neurons look like at that scale? What the unhealthy neurons look like, and unfortunately, we can't even dichotomize into healthy and unhealthy. There probably has to be some sort of quantitative way that we can assess, for example, cortical excitability. So broadly speaking, you can consider epilepsy as a problem of cortical excitability, specifically hyperexcitability, or sometimes you might say paroxysmal hyperexcitability, but even then that's a little bit misleading because there probably is underlying hyperexcitability and underlying compensatory mechanisms that are at work all the time.
So by paroxysmal, you mean regenerative or something that could take off by itself? Is that
yeah, by paroxysmal, I mean the hallmark of epilepsy are seizures, and those are paroxysmal in the sense that we can't predict them often. They sometimes have triggers, but they often happen for no reason at all. And so we call them paroxysmal, but clearly events of hyperexcitability. But for most epilepsy patients, they live most of their life without having seizures. They might have 3 minutes of seizure every month. Now that 3 minutes can be extremely disruptive to their lives, but for the rest of the month they're fine. We just don't know when that 3 minutes is going to happen. Yeah. And so you think about that time in between, is the patient completely normal? Well, I think all of us are going to agree that the physiology is not completely normal, even in that, quote, interictal period. There's comorbidities to epilepsy, but even strictly speaking from the epilepsy standpoint, probably there are compensatory mechanisms that might even be in relative overdrive in that interictal period in order to stop these paroxysmal events of seizures. And that's
if you could see that happening, you know a lot. I mean,
exactly.
For one thing, because you can get a lot of data about that time, because there's a lot of that time.
That's right. There's a lot of interictal data, and it's much easier than what we do right now clinically, which is even on the scalp. We admit patients all the time for EEG into the epilepsy monitoring unit, and we say, we won't know more until we capture seizures. And I would say there's a lot of patients where they come in and they say, Dr. Lundstrom, I usually have seizures all the time, but now I'm here in the hospital and I'm just not having them. What can we do? And I'm like, well, we'll try to sleep deprive you and so forth, but it's still a challenge. So that's where we're at. We're still waiting for seizures. Yeah. The things that we don't want, the things that we want to treat, we want them, at certain times so that we can move the process forward. It's not a highly refined system in that regard. The idea that we're going to induce bad things in order to stop them in the future, there ought to be a better way.
There's no EEG indication at all in this interictal time that there's anything odd about this patient's brain at all.
Well, there can be. There can be these interictal discharges, and so we do see those in a majority of patients. And that will help us know that they do have seizures, and it will give us a general ballpark sometimes of the area, but it doesn't give us enough information sometimes. So that's the challenge, is we want more information than that. And furthermore, these interictal discharges, which you can see, it doesn't give us information about whether the current therapy we're doing is sufficient. So the way that we currently manage these epilepsy patients is that we treat to failure. We treat, we guess, and then if they fail, we change. And it would be nice to know that we had an adequate treatment before they fail again.
Yeah. So I guess the hang up is that, I guess if I wasn't a neurophysiologist, I'd say, well, looking at the electrical activity isn't the way to go. We should be looking at something else, gene expression or something like that. Because this patient still has the disease in the in-between time.
Or you might say, maybe we just need to look at some different aspect of the EEG.
Yeah.
And that reading the raw EEG might not be the best way to go.
Or maybe if I think what you described is so much like the way it used to be with my television set. Television set doesn't work. I take it to the television set fixing guy.
Mm-hmm.
He turns it on, it works perfectly.
That's right.
And then the minute it's out of the shop, it goes wrong.
That's right.
But the television fixing guy he had diagnostics. He would inject signals into different parts of the television circuit, and he could see, latent, the incorrect signals that didn't cause the TV set to not work. So what about the possibility of sort of challenging the brain electrically, especially with this looking for excitability?
So you could reasonably view an interictal discharge that we use all the time clinically as a passive biomarker. It has its problems, but it's a passive biomarker of the seizures that intermittently happen. The other alternative, which has certain advantages, is you could say an active biomarker or a stimulation-based biomarker, where we're going to provide stimulation in some protocol or some fashion, and we hope to get back information that's going to be helpful to us. And so there has been a lot of work recently with things like cortical evoked potentials and how do they change with stimulation over time, and how does that help us better understand epilepsy. Sometimes we even induce seizures, although that has its own problems because seizures always carry the risk of morbidity. But the challenge still remains is how can we understand the signals that we get back, and what's the best way to understand them? And that's where some of my basic science research has started to come in, where the idea is that maybe the best thing we should look at is not the initial response we might see to stimulation, but it might be how that response changes to stimulation over time. And this, we think, is related to fundamental mechanisms of single neurons like spike frequency adaptation, where the action potentials slowly, the rate of the action potentials changes slowly to a constant stimulus, this adaptation. So that being a form of gain control that might have relevance even for epilepsy patients where the brain is trying to control excitability.
So in that case, those kinds of mechanisms would be constantly cranked up and holding down the excitability of the cortex all the time. So you'd find the brain in a state of negative feedback, overactive negative feedback, as well as overactive excitability. And you could detect that maybe because you understand so much about where it comes from at the cellular level.
That's the hypothesis our lab is working on, is that there is this adaptation, these adaptive mechanisms that are at play, and they are probably ramped up at least around the area of focal onset. And there may be signs in the EEG that that's the case.
Like what?
Well, so what we've been looking at recently, and a special interest of mine is related to the slower aspects of EEG. So broadly speaking, in EEG, we typically look between 1 hertz and 70 hertz. That's sort of broad, narrowband EEG that's been well accepted for quite some time. Maybe about 20 years ago, there was increased interest in high-frequency EEG with high-frequency oscillations and so forth. And there's a lot that's been learned about that and its potential as an interictal biomarker. But I'm particularly interested in some of the lower frequencies, perhaps between 0.01 hertz and 1 hertz. And that interest stems from some of my interest in these mechanisms of adaptation that come from the single neuron level, where it seems that we should see some signatures in that frequency range. That is to say, below 1 hertz or 0.01 to 1 hertz.
So can we record EEG at those really low rates, and we don't have to worry about like electrode polarization or something like that messing up the...
You do have to worry about some issues. There's no doubt. And amplifiers are also sometimes an issue. The amplifiers, broadly speaking, even the clinical amplifiers are better now than they used to be for recording what sometimes people call DC potential recordings or recordings that don't have, I guess you could say, a high pass filter setting on it, because that is an issue. So there are some aspects that are important to consider.
So this, maybe this is kind of naive, but my understanding is that the really low frequencies get accentuated in sleep.
That's right.
And so is sleep a good time to look for this?
It is, actually. We think that sleep is actually a really good time. There's evidence that it's sometimes it's even a good time to look for other things too, like HFOs and high-frequency oscillations and interictal discharges. But we do think it's a good time to look for some of this activity in part. For example, what you see in sleep is oftentimes rhythms kind of in that 0.3 to 0.5 hertz range that are related to up and down states. And the transition between up and down states is probably related, at least in part, to these adaptation-related mechanisms. And so, yes, I think it's a great place to look.
So there's an opportunity to zoom in because a lot's understood about single neuron activity during sleep, especially the synaptic currents that are flowing in those neurons because people have done intracellular recordings. And then the synaptic currents are probably what's contributing to the EEG. So is there a real deep understanding about what the EEG means at those low frequencies, at what the cellular mechanisms are at those frequencies?
I don't think there's a deep understanding. I still think there's a gap. The gap between sort of the high-level EEG and sort of some of these lower-level neuronal recordings, as you mentioned. But I think it's an area that's promising.
But like at the low frequencies part that they seem to be enhanced during sleep, wouldn't those be associated like with a resting network? Would that be something that then you're just looking at the fundamental network connectivity and then everything else that is higher frequencies just mounted on top of that, or different permutations of that network?
Well, I think broadly speaking, there is this idea of, for example, cross-frequency coupling and how lower frequencies tend to modulate higher events. And I think there's a lot to be said for that framework. So I do think that a lot of higher frequency events are modulated by low frequency oscillations, which I think probably just speaks to the importance of the lower frequencies in better understanding how excitability is controlled.
Mm.
You can make a distinction between how much excitability there is at any given time, maybe, and how well excitability can be controlled in general. So you want a brain that can shift between levels of excitability. A brain that is completely stable is boring and unhelpful. So you would like a certain dynamic range. If your speakers only give you 440 hertz, you're going to be stuck with A.
Well, that's a computational idea of what adaptability is, from the level of an ion channel to a sensory neuron, right? That keeps you on a dynamical range and keeps you, like moving from a dark room to outdoors, and that allows the same detectors to give you the same ratio in contrast. And you could think that that is implemented at the network level. But also a healthy EEG, I don't know, tell me if I'm wrong, has like a power law spectrum, right? I mean, that seems to be one of the ideas that I read is that is because you're engaging fast and slow networks. They are like all together, right? If you just had a single oscillation, that's not good. And if your brain is running at 60 hertz, that's not good. But when you have coupled networks across the entire brain that are going, and then each other affect each other, it's a natural process, mechanism that gives rise to power law networks. And then how do you think that correlates to a brain that is okay or is not okay, and can be a biomarker for epilepsy or any other disease?
There is a discussion still about the form that EEG signals tend to take if you look at them in the frequency domain. If you're talking about higher frequencies, so let's say above 100 hertz or so, there is no doubt that it tends to have a power law form in the exact contribution of the low-pass filtering characteristics of the membrane versus noise and so forth. There's still active discussions about some of that. But what you tend to see in EEG is that once you get lower on that frequency range, so lower than 50 hertz perhaps, but especially lower than about 10 hertz, things change. You're looking at a different regime. It's not a power law still. You tend to see often some flattening there. And so we think that the mechanisms underlying that particular frequency range of the EEG is probably a little bit different. And from our perspective, perhaps more important, if the question you're trying to ask is, what governs this dynamic range ability of the brain to function? So not necessarily the question of what is the activity at this exact moment, but what is governing how the activity is being controlled.
And in fact, these slow oscillations, if they have anything to do with what we've seen in neurons, are really shifts between an excitable state and an inexcitable state.
That's right. So almost by definition, then, it's this shifting between high and low, and maybe to some extent even determining what excitability is.
So one last thing I wanted to ask about that low frequency stuff is, at least it used to be thought that low frequency, the low frequency signals are more correlated over a broader area in the cortex, and high frequency less so.
Yep.
So low frequency things are global and high frequency things are local. So I don't know what that means in terms of mechanism exactly, because I don't know what makes some signal global in the cortex and some other signal local. But if we're looking for a place where excitability is really high, and we're looking at the global things, then I guess we're looking at a deviation from a global signal. Or
I think that's a good point. There's no doubt that it does seem that the signal that the postsynaptic potentials that give rise to the lower frequency parts of the power spectrum come from larger areas of cortex. I think I guess I wouldn't necessarily say global right away. I would just say larger. And then I completely agree that then what you're looking for are differences between areas. And there's a number of studies, even with high density EEG and some of the source localization that they've done, that the localizing power of these focal changes of slow activity can be similar to the high frequency. Now, so how can that be? Well, I think there's a lot of details that go into some of these aspects, but I don't think it's necessarily the case that there's that you can't localize with the lower frequency activity.
I don't want to let you get away without talking about treatment by stimulation, because electrical excitability is the problem. Then why can't we just apply a field that makes the cells less excitable, or activate an inhibitory pathway, or
I guess the problem is the brain isn't as simple as we wish it were, at least when it comes to these kinds of approaches. So transcranial direct current stimulation probably does, if you apply cathodal stimulation to a focal area of the brain, increase the spike threshold of action potentials by probably half a millivolt or so. So a very small amount. So it does probably do that. There's some discussion about that, by the way, but that's a possibility. The brain stimulation that we tend to use most often is invasive brain stimulation, like deep brain stimulation, DBS, or responsive neurostimulation, RNS. And these are all, relatively speaking, high frequency, very short pulses. So on the order of 90 to typically it's usually between 90 and 160 microseconds. That's what's most often used. So these are very short pulses, and the frequencies are typically in the 100 to 200 hertz range. So fast. How do they work? Nobody knows for sure. So that's an area that's still left open for some debate, speculation, and further research.
The ionic fluxes, that's the last thing I've heard. I mean, you make fluxes of ions in the extracellular space, and that's how you regulate that instead of affecting haptically the membrane. So, I mean, it could be.
Yeah, I think most people agree that for most stimulation protocols, you are causing some neurons to fire. So it's not that you're silencing neurons directly.
Yeah.
So I think most people agree on that. And once you get past that, it becomes a little tricky.
Right. But you might think it would be spatially specific. So if I had a epileptic focus in the forebrain, in the frontal cortex, then I should stimulate some place that projects to the frontal cortex. And if it's in the occipital cortex, I should stimulate a different place that projects the occipital cortex. Is there any pattern like?
Yeah, absolutely. So we use that kind of rationale all the time. I'll give you one example. For deep brain stimulation for epilepsy, we tend to stimulate in the thalamus. If the seizure tends to be in the limbic circuit, then we tend to stimulate the anterior nucleus of the thalamus, which has strong limbic connections. Yeah. If the seizure, for example, tends to involve the perirolandic or the primary motor cortex, we might think about the centromedian nucleus of the thalamus. If the seizures are in the posterior quadrant of the brain, then sometimes we tend to think about the pulvinar nucleus. Now, there's a lot that's unknown about a lot of those regions, and especially the pulvinar, about exactly where you stimulate. But that kind of thinking is certainly something that's considered.
All of those are interesting because they are not the most direct sensory relay, or anything like that. These are these thalamic nuclei that are considered to have indirect effects, like cortical effects or something. Is that a pattern?
No, I don't know. It's true. But that's I think what's been done.
Well, but also you can think about loops, right? I mean, parts of the brain that are involved in loops in the brain at multiple scales, right? One of the ways that you can generate these kind of like slow waves, and you can imagine this is like you have a neuron that generates an action potential, connects to another one, and then feedbacks right away. Then another axon that is a little longer contacts to another neuron, and so on. And you make a chain that provides feedback in 1 millisecond, 10 milliseconds, 100 milliseconds. And that can be generalized to an entire loop. Like if you're in the thalamus, well, if you go to the cortex, it could be the striatum and then cerebellum or whatever. As long as you're in the loop, your pulse will get propagated and feedback to you over all the scales of interest.
Anywhere out of work.
It could be, but it could be in the balance between those that you have a transition between what controls excitation and inhibition, right? or locally. But these kind of that's what generates these kind of correlations at longer scales, and can generate this low frequencies and also the power law structure at higher frequencies. So as long as you sit not I don't think everywhere, but
So what do you think are the prospects for that? Do you think there's better things on the horizon, or is stimulation going to work out really well? Do you think,
we occasionally see patients for whom stimulation works out really well. It's unknown to what extent, if we optimize stimulation and its vast parameter space, we can improve any given patient yet. We need a excitability biomarker to help with that. It's really unknown to what extent stimulation can become more of a curative therapy. Right now, it's a non-curative therapy where seizure freedom is pretty rare. It's typically prolonged periods of seizure freedom are sort of in the 5 to at most 15% range, and even that's typically time periods of 1 to 2 years and not forever. So there's a lot of unknowns, but every once in a while patients come up where changes seem to be long-lasting and not dependent on continuous future stimulation, and it seems like some sort of plastic change has been made to the brain that has led to very long-term seizure freedom. So we'll see. But again, I think a lot of this, unlike in movement disorders where we have an immediate readout of the effects of stimulation, we need some way to know what we're doing more quickly than waiting for further seizures.
Brian, thank you very much for joining us. Seems to me bioelectronic medicine is forced on us by the brain, by the structure and function of the brain. We don't have any choice. We have to go for it and try to master that. And
I guess I should add, maybe on a note of more optimism, is that we've been talking about epilepsy, but there's a lot of brain problems, from addiction to psychiatric issues and a lot of other neurological dysfunctions that are probably related to issues of excitability. Mm. And so although we've been talking about this in terms of epilepsy, in a lot of ways this could have broad application to a lot of other
like depression.
Yeah. Exactly, yeah. For example.
Mm. I mean, that's it. I mean, talking like that, you're saying that it takes a long time to see if it's working. There's some parallels with depression there.
That's right.
It takes a while.
That's right. And of course, the goal is that we just need to better understand, I think, this relationship between what happens at sort of single neuron levels and how adaptation happens and maybe how that happens over time. And, we've talked in other settings about fractional dynamics and power laws and how these theoretical concepts, which can be somewhat tricky to understand, might be relevant to understanding how the brain works, what we see on EEG, and hopefully then long-term better treatments for.
And our best prospect for filling in the gap that you were talking about, the spatial scales gap, is actually theory that would allow us to make the connection between those things.
I think it's an area where we need more theory, we need more modeling, and continued experiments. Yeah. I don't think that, unfortunately, AI is going to solve this problem for us without these components. And I think it's a good time to be involved in a lot of these aspects. There's a lot of exciting opportunities and a time not to neglect maybe some of the, quote, hard work of doing theory.
Right.
Well, that's a good thing to end on.
That's a wrap.
You could say that. So, thanks a lot for joining us, Brian and Fidel.
Thank you, Charlie.
Thank you, Brian. Very much.
Mm-hmm.
This has been Neuroscientists Talk Shop.